Hello, everybody, and welcome back to our 50th
Yeah,
episode of
50!
Fossil Friday Chats.
Woo!
Yay! Yeah, we actually made 50 episodes. Thank you guys for being with us all this year. I'm Brittany Stoneberg, the Outreach and Communications Coordinator for the Western Science Center. With me, as always, through 50 episodes is Gabe Santos.
Hi, everybody. I'm the one very, very patiently waiting to watch the season finale of WandaVision after this, so, yeah.
He's been so patient, everybody. And joining us today is our friend and paleontologist, Yara Haridy. Yara, good to
Yay!
see you.
Hi, thanks for asking me to be here.
Absolutely. I am very excited to hear all about bones.
Yeah, thanks for giving me a reason to actually, do my makeup today and get out of bed. It's much appreciated.
That's what I use these. That's what I use these. This is when the lipstick gets used.
Yeah, right? I mean, it's probably like perfectly dated. It's like this lipstick is that day, and then everything else is just like, and I'm wearing pajamas all day.
Yeah, this is the day when I wear, like, button-ups and I can, like, actually put, like, moisturizer and stuff on my face because I'm going to be on camera. Otherwise, I'm just, like, I can go
Eh.
from bed to desk within a second.
Very very short commutes nowadays. So if you're not familiar with our friend Yara, she is a science communicator and science at the Museum for Naturkunde in Berlin, Germany. She's an expert on teeth and bone evolution, living things, and what they're made of. Yara is working on several projects which encourage equitable fieldwork practices with the aim of decolonizing paleontology. During the pandemic, her research has been put on hold. Instead, she's been helping to build a museum.
That was an amazing intro. Thank you so much,
You're
Brittany.
welcome. And also a reminder to all of our viewers, as always, if you have questions for Yara, go ahead and put them in the chat. We'll have our brief Q&A session after Yara's presentation. So if you've got any questions, and I expect that you will, because this is going to be a really interesting talk, just go ahead and put them in the chat. So Yara, whenever you are ready, tell us about some bones.
Awesome. So like Brittany said, I have a diverse field of interests. It's gonna seem a little scattered and it's gonna seem like a little crazy, but I also welcome literally questions on all fronts, from paleo art to the specifics of bone cells to teeth. And I think the funner part of this talk will be the tail end, where you guys can ask me all kinds of questions and watch me struggle to answer them. So let's get into it. Skeletons and bone evolution. So basically, a little bit about me at first. I didn't get into paleontology in the most traditional of ways. I basically started. Yeah. Oops. Can you still hear me, or? Welcome back, everyone. I see you're still interested in bones. So I'm basically going to talk a little bit about everything that interests me, and that way everyone can get kind of a appetizer plate of everything that I'm working on, and that way we can chat about whatever, and you can ask me questions whether it comes to modern bones or fossils or how to get into paleo this way. Anyway. So, a little bit about me. I didn't really get into paleontology in the most traditional way. I didn't even start knowing what fossils were until my very last year of university. And basically, I was, studying towards pre-med, much like many immigrant children. I was focused on going to be a doctor, making the family proud. We're going to do this, do the science thing, and the science thing was only medicine, to my knowledge at the time. So I basically wanted something to set myself apart from all the other applicants and wanted to volunteer at anatomy lab. And the only anatomy lab on campus, oddly enough, was a paleontology lab. So that's how I got to actually volunteering at this lab, which worked on very small reptiles. And these are Permian reptiles, but not the kind that not the kind, not like modern reptiles. They were very much more simple and small. And so on the side here, you can see me holding like a little ulna, which is your arm bone, a part of this reptile's arm bone, just to kind of give you guys the scale that I was working on. And this was the much larger scale that was present at this lab. So I didn't even know that real people worked on dinosaurs. I didn't know that people actually really dug bones out of the ground and put them in museums. And I don't know why. I never made that connection that the museum things that are in the museums are actually filled by real life people and real life scientists. That was never really explained to me. That was never something I was exposed to. And that's partially, I think, why I'm really interested in science communication, because I feel like I kind of missed out. I feel like if I had known this stuff earlier, I would have gotten in earlier. There are so many cool fossil camps and fieldwork opportunities for younger students that I could have gone in on. I just didn't know. So anyway, enough about those woes, more about fossils. So I got really interested in anatomy through basically this ancient anatomy, and I asked all these dumb questions. And there are no dumb questions, but some of my questions were real dumb. Some of them were, why are these teeth bigger than this? Or why are you even looking at this? And I kind of didn't understand why paleontology was asking these questions, why anyone would even want to study Permian reptiles. But I got this explained to me wonderfully in my experience there, and I was hooked. I was hooked on these little reptiles. I was hooked on their bones. I was hooked on asking these big questions with little bones. And that led to much bigger experiences, like this picture on the side here. So this is the last excavation I was on in October 2019 in the before times, where everyone could be still near each other and digging up really cool dinosaurs. This is a giant brachiosaur humerus. I believe it's like one of the largest ever found. And we pulled it out of Utah just a couple years ago. And it was an absolutely amazing and addictive experience to pull this monster out of the ground and then realize that the bone on the left and the bone on the right are actually made of the same cells, or were when in life, when the animals were alive, they had the same cells, they had the same processes. They are all connected. And they're connected to the bones that are inside us. And that's kind of what drives me. Just these little connections that lead to extremely different morphologies. So again, like I said, I'm interested in science communication. I'm really sick and tired of textbooks kind of being the go-to way of explaining things, or PowerPoints, ironically, being the way to actually explain things. So one of the things I'm trying to do is bring specimens to people. I set up a kind of like touch and talk table at the Museum for Naturkunde in Berlin, where I got to speak to people about the difference between antlers and horns and what makes those different, like what's different about your bones versus your hair. Another project I'm working on with Brian Ng, and that is funded by the Palace Association, is we are building a velociraptor puppet that we can actually bring to museums and bring to parks, hopefully when we can actually interact face to face. But we'll actually make, hopefully, some parts of this puppet publicly available so people can build them at home, and then we can talk about why you're building this animal this way and that way, and what about biomechanics? Why are animals built the way they are? Why are bones placed the way they are? Because I really do believe in hands-on experience and hands-on learning. I think the best way to learn about what a femur is to give someone a big, like, cow femur and have them hold it, feel the weight, explain to them how much muscle mass is actually around bones. Anyway, so I'm excited about bones, excited about skeletons. Let's actually get into it. One of the other things that I do want to kind of invest my time in the future is my bare bones blog, which is kind of why I called this talk The Bare Bones of Skeletons. A lot of these topics seem really convoluted, but when you strip them down to the bare bones, I think it can be really understandable. And the problem I find with things like histology, odontology, the study of teeth, osteology, the study of bones, paleopathology, the study of ancient diseases, it's really hard to find basic information explained simply so that people can catch up and kind of figure out what's the cool stuff that's coming out today. Finding the basics is hopefully what this blog is going to be about. Just simple, what is odontology? What is the study? When did it start? Why, that kind of stuff. So that's a blog I'm working on, but it hasn't gone too far just yet. And lastly, again, my love of bones, skeletons, and morphology has brought the game Guess the Skull to Twitter and Instagram. So for anyone that is watching here but isn't following me on Twitter, you're missing out, because there's a weekly game where I basically post a skull picture, and then about 24 hours later, I'll reveal what the animal is and talk a bit about the animal and sometimes how the bones actually influence behavior or influence the way the animal is shaped. And it's really interesting to see other people's guesses, because you can start to see how people will use week to week knowledge to kind of explain, oh, this animal has this many teeth, so, it probably eats this. Or this animal has blunt teeth in the back and sharp teeth in the front. What does that mean? And you can see people helping each other out in the comments. It's a lot of fun. I suggest you guys join. Anyway, so I was asked here today because I'm apparently a scientist. So what do I actually study? Well, it's hard to explain because it seems like the things are not very connected. I study several what seem to be very disparate things. I study bones, I study teeth, I study disease, I study cells, and I promise you they're all connected, and I hope you think so too by the end of this. So one of the first things, my master's in Toronto in Canada, was on tooth replacement. Not in Smilodon exactly, but this is a really good example showing how a juvenile Smilodon will replace its teeth over a lifetime. So I always use this example when I'm teaching tooth replacement, because people don't understand how you can actually go from a young tooth to an older tooth and the replacement process. And what's really interesting is, mammals, Smilodon, and ourselves do this only once, while things like sharks and reptiles do this throughout their lives. And we can actually glean a lot of information from tooth replacement, but we won't get into it too much today. If you guys are interested in teeth, tooth replacement, and weird tooth evolution, you can ask me about it at the end, and I can get into it. So one of the ways I did study teeth, and I study bones today in the same way, is histology. So basically, these are all modern animals. So what you're seeing here is an iguana, python, alligator, blah, blah, blah. What you're seeing is a stained animal, and then the slices. So basically, the animal gets in parts of the animal gets embedded, and then we slice them, put them under the microscope, and then we can study the cells. Now, what usually blows everyone's minds is that we can actually do something very similar with fossils, and we'll get to it in a second. So that's teeth. I got interested in the cells that make teeth. I got interested in the bone that surrounds teeth. I got interested in why they have all these different shapes. Where do they come from? What did they evolve from? But teeth don't exist in a vacuum. They don't exist by themselves. They exist in our jaws, and therefore you have to kind of understand bone as well, because they move together, they grow together. And my understanding of bone was very shallow until the start of my PhD, which was going to be on bone cells. So all of a sudden, I had to understand what is up with bones. So in this picture, you can see a couple of vertebrae up at the top left. But what's obviously taking up most of the picture is these two femora, these two giant femurs, leg bones. So you can see my thigh, my hip to my knee is my femur, And then this sauropod femur that is on the Dinosaur National Monument wall, which I got to take some measurements and kind of check out in 2019 for some pathologies, which we did find. So it's just a fun, striking image to see that these two bones that are likely very, very similar in cell structure, in muscle attachment, and a bunch of things, but the size is just ridiculously different, and yet the cells are doing very similar things. So I ask, why are humans not a pile of goop? Oh, sorry, not this image. Not a pile of goop. People don't really get what I mean by that, but when you think that animals were simple for a very long time and didn't really have a hard part to them. They were squishy. Most life was in either the form of bacteria or archaea or other invertebrates that didn't have exoskeletons. This was for a very long time. However, as soon as hard tissues evolved, like here in this example, if you notice at the very end, there's little hooks. So this is probably not the best example for a squishy animal because you can see they have teeth, not true teeth, but basically these little hook-like appendages for attaching. These bloodworms are a perfect example for why hard tissues are important. Hard tissues have basically changed life on Earth, and I don't say that lightly. If we expand our definition for skeletons, we can understand why. So for example, skeletons are super important because when you have a squishy structure like muscles and tendons and all that, they can only push so much against each other. But if you push against something hard, all of a sudden the efficiency and the actual movement that results out of these muscle contractions is much bigger and much better, and you can actually move an animal in many different ways. And that's probably the dumbest and simplest way to explain biomechanics, but it is what it is. I mean, you need some amount of hard tissue to act against your muscles. And if you expand the definition of skeletons, like away from bones, then skeletons are actually quite old. It's not really a new phenomenon. This is a sponge skeleton, so while it's very different in composition, it's made of silica, so like much closer to sand and glass, and they don't use it to move. They use it more for protection and separation. I am not a sponge expert, sorry for anyone that I am offending, but basically skeletons have been around for a very long time. And a better example is, of course, exoskeletons. Skeletons on the outside that are not made of bone. They kind of use them in the same way. Exoskeletons are not just on the outside. They actually have flanges that go inside the bodies of these animals, and they do the same thing as bone. They use them as a counteraction to muscles. So that's why these animals are not piles of goop. That's why today you're not a pile of goop. It's for efficiency of movement. So back to bones. Then if a bunch of animals have been doing it for a long time, who cares? Why are bones special? Why do they matter at all, and why am I studying them? Well, bone tissue is quite different. It's alive. Unlike silica, so for a sponge, they just kind of extrude this material out and then it's dead. Same thing with crabs, they will put out their shells and then when they grow, they have to molt the shell and then grow another one. So the tissue itself is dead, but bones are a live and active tissue. Excuse me. They also continue to heal. They continue to grow. They react to every single thing you do every day. They will change depending on your diet, depending on your exercise regime. If you want to do something good for your bones, you should actually weight lift. You should, make sure you have a balanced diet. They're this active and continuously changing tissue, which is very different than most other skeletons. So how do we know that they are this active and continuously living tissue? Well, we study cells, and they have living cells inside them. They are surrounded by living cells. Bones in general will actually maintain a record of what you've been doing all year or through your life, through the many years. So, for example, one of the ways we know how old a dinosaur is or how old an animal is we'll count the rings in bones, the same way you would count rings in trees. Now, I'm particularly interested in the smallest micro unit of bones, which are the cells that they're made of. And I'm mostly interested in why bones are the way they are. Why are we not covered in exoskeletons? Why are our bones not made of silica? Why are these cells more advantageous than any of these other simpler or older versions of skeletons? Why did vertebrates reinvent the wheel? Why didn't we just have skeletons made of sand or glass? So then I have to look at the cells, right? But if you're looking at the oldest cells, you're looking at some old fossil cells. How does that look like, and how do we study them? Well, I do this through histology, the same as I showed earlier for teeth. Basically, you slice up these bones. This is a slice done by the MOR, I believe, and it's of a theropod bone. So what you can see here is all the little black dots. They're not true cells. They're the spaces where the cell used to live. And that little Y formation you see in the blue circle, that's where a vessel used to live that would supply nourishment and blood to these cells. So there's this incredible detail of evidence in fossil bone that actually gives us information on how this bone used to remodel, how it would heal, how it would make itself, break itself down, and then make itself again. All of that is recorded in fossils. And to me, this probably is what really hooked me onto paleontology, because while the shape of bones is interesting, while finding bones in the rocks is pure magic, looking into the bones and being able to be like, oh, this animal had a disease and then healed, and then it grew some more, and then it had a really rough year, having that level of detail is ridiculous. And I didn't understand how it could be preserved this well. And better yet, now we can actually see it to the cellular level, which is pretty crazy. So let's talk a little bit about bone cells. So obviously, to understand bone cells in fossils, you really have to understand bone cells in modern animals. So this illustration here is done by a dear friend, David Mazursky from the University of Toronto, and it was done for a paper that's hopefully going to come out soon. Anyway, so what you can see here is the vasculature on the surface, these osteocytes, and then the osteocyte lacunae. So the osteocyte lacunae is just the cave that the osteocyte used to live in. And basically, these cells lived there, and they would talk to their neighbors. They would send out their little tendrils, and together, all these cells sense pressure. So if you are putting a lot of load on your bones, they can sense a lack of pressure. They can sense an increase in minerals. So say, for example, you have a lot of minerals in your diet right now that they can use up and store in your bones, or you have not enough. For some reason you have a dietary deficiency. All of a sudden they'll tell each other, hey, take a little bit off your wall and send it out. Take a little bit off your wall and send it out. And that's how we change our mineral metabolism. So bones are very alive. They are involved in our hormones. They are involved in growth. They're involved in a lot of different things, and we're discovering more and more every year. But back to fossils. How do we tell what these cells are doing? And have they always done what they do today? And that's basically my whole PhD in a sentence. Do the cells that exist today, did they always do what they do today? So here in C and D are two thin sections that are from my museum. They were done early in the, I believe, 40s, but I got to re-image them. And what you see on one side is basically this orange glop with a bunch of lines. And on one other side, you see a bunch of orange glop with a lot of black dots and lines. So the black dots are the cells. So you have some bone that has cells and some bone that doesn't have cells. And that's pretty crazy when you think about how much these cells do and how many therapeutics are based on these cells today. So sometime in the past, in the far, far past, 420 million years ago or so, early jawless fish, or our very early bony-ish ancestors, they didn't have these cells. but they seem to be doing just fine. So why did we evolve these cells, and what do they do for us? Why have them in the first place when realistically having all these little holes in your bone actually makes the bone less dense and possibly less strong? So these are some of the questions I'm asking and trying to address during my PhD. Now, the star of the show here is the osteocyte, the main bone cell, the one that preserves the best shape in fossils. So that's what you're seeing in that little image down there, the osteocyte. However, there are many other bone cells that definitely do not preserve or almost leave no trace in bone at all. So the osteocyte we talked about, the osteoblast is the bone building cell, osteoclast is your bone destroying cell, and osteoprogenitor are ones that line the bone that are basically precursors to osteoblasts and osteocytes. But, like I said, out of all four of these, only one really preserves. But we need to know the full story. We need to know what the other ones are doing. We know from fin sectioning that somewhere along the line, somewhere at that red circle at the bottom there, osteocytic bone appeared. But everything before that was anosteocytic, meaning it didn't have osteocytes, but presumably had the other three versions because it exists. We know that the animals grew, we knew the animals made bone, so the other two cell types must have existed by proxy of the animals themselves existing. So another thing I'm trying to approach here is basically trying to understand these cells by understanding how the bones are shaped. Now, stay with me. This seems a little crazy here, but what I'm trying to say here is that on the right, you can see a bunch of maladies and pathologies in bone. So the orange one, the bone is broken and is healing weird. Sometimes you get a bone that splits, or it has a congenital deformation, or you have diseases where there's too much bone building or too little bone building. And all these different diseases, all these different pathologies are basically experiments that have gone wrong, but they tell us about what is going on in the cells. Now, I know that this sounds like I'm making connections maybe when they aren't there, but paleopathology is the study of ancient diseases, and it's kind of started off as my pet project, where I just was interested in the fact that we can find diseases in fossils. However, it turns out that they actually might be the clue to understanding what these early cells were doing. So stay with me. Paleopathology, the study of ancient diseases. Some of the earliest versions of diseases we know, at least in humans, are cancers and bone cancers because they preserve so well. So 1.8 million years ago, there was some poor early hominid with an osteosarcoma, and this was published in 2016. Pretty cool scan. I'm pretty sure it's publicly available for anyone interested. And then we published in 2019 the earliest cancer in a stem turtle. So this is what you're seeing here is a femur, and it has a big growth on it. And that's also publicly available, and we can talk about it more if anyone's interested. Now back to these connections. Here's a really good case study to kind of point out what I'm trying to talk about here. When a bone is perfectly formed, let's say you find the most perfect T-Rex skull, okay? You actually assume that, yeah, okay, it has all the cells that we have today, it made them the same way, because there is no null model. There's no option to see that the bone wasn't working. And so when it doesn't work, you can tell this part is missing. So therefore, there's this when you find a perfect fossil, there's kind of a black box around the cellular mechanisms behind it, because we don't know what is in there, we don't know what makes it tick, because everything is working perfectly. And therefore, we assume everything is being built the same way it's built today. So here's a good example to kind of point out what I'm talking about. So here's a perfectly formed varanopid vertebrae, and it just looks like a nice, pretty vertebrae. I mean, it's 280 million years old, so that makes it a really pretty vertebrae. Not crushed, gorgeous, Great, we scanned it. Here's another one. Same animal, not the same individual, but the same animal. There are two vertebrae in this spinning model, and they don't look as happy. They don't look as perfect. And it's from the same locality, and it's really well preserved. So it's not crushing, it's not taphonomy, but they're actually fused together. Not only are they fused together, but they have all this buildup of bone. And sometimes you can't tell this from the surface of the fossil, because here you can see what the fossil looks like externally. Really, really smooth bone, doesn't really look diseased. But then when you scan it, you can see that there's all these internal, basically, it looks like Swiss cheese, let's be honest. There's overbuilding, it's too thick in some areas, and there's way too much hollowing in other areas, and it's fused, which is abnormal. So what's going on here? Here you can see I'm pointing to some of the holes. I'm pointing to where it's fused, where it shouldn't be. And these holes basically tell us, or this combination of symptoms basically shows us that there was a metabolic bone disease. Metabolic bone diseases are basically when there's a problem with how your bones form and how they are put together. And remember, this is a 280 million year old specimen and animal. So being able to narrow it down to metabolic bone disease is pretty good. So we know that there was some kind of miscommunication with the cells in this animal. So because there was a pathology that happened, now we know, okay, we're going to look at the osteoclast and the osteoblast, the cells that build bone and the cells that destroy bone. And because of this pathology, because of this mistake that happened, we know that the osteoclasts, the ones that destroy bone, were upregulated, and the ones that build bone were downregulated. And this kind of helps us look behind the black box. And therefore, we know that these cells communicated with one another. Now, obviously, these cells communicated with one another because animals grow and heal and change. But if we find metabolic bone disease in even earlier animals, that is actual proof or evidence towards these communications existing a long time ago. And we can't assume that these communications always existed because there were so many tissue experimentations early on. All these tissues don't exist today. So we actually don't know when these communications between these cell types were cemented. And so it's interesting that paleopathology can actually help us understand bone evolution and therefore help us understand why our bones are built the way they are. And in the end, help us understand why we're not just a big pile of goop. So some other things that you can ask me about or talk to me about, especially if you're interested in paleopathologies, is fossil cancer. This is a beautiful illustration done by Brian Ng, illustrating the fossil cancer that we discovered in 2019. But also, we are so lucky and we are living in the golden age of paleontology. And the first confirmed case of cancer in a dinosaur happened just last year by some colleagues at the ROM and at the Chirel. So lots of cool paleopathologies coming up in the pipe works too that I know about. So get excited about paleopathologies. Other things you can ask me about is, well, I'm basically an expert at finding dead things. I've been like that for a long time. I'm interested in taphonomy and where and how to find them. So if you're ever, people always ask me, Yara, how do you find so many bones? Or how do you stumble on all these dead animals? So if that's something that you're interested in, let me know and ask away. And of course, I'm now on leave from research because of COVID. So I'm working on a Dilophosaurus exhibit with Brian Ng. And so here is a picture of me helping sculpt a Dilophosaurus arm, which really has no business being that large. That is my cat on my lap. So if you're interested in paleo art and the sculpture process and how to design any of the exhibits, you can try to ask me. If I don't have the answer, I will point you to Brian, who is the mastermind of all of this. And yeah, and if you want to talk about decolonizing paleontology and how some of the field work that we have planned is being planned in that direction. You can also ask me about that. Like I said, many diverse interests. So yeah, ask me about your bones.
You know what I want to ask about?
What do you want to ask?
Sobia.
Oh, she's so good.
She's so cute.
That...
She's adorable. Oh, thank you so much, Yara.
Oh, you're very welcome.
Yeah, that was a fantastic talk, especially because, like, for paleontologists, we talk a lot about reading the rocks to, like, deconstruct the past, but you literally can read the bones, and that's such a cool thing to do.
It's pretty fun. I mean, if I didn't have histology, I don't think I would have stayed in paleo, because it's like all of a sudden being able to see color. It's like another dimension to your whole field. It's pretty amazing.
Awesome. Well, go ahead, Gabe.
I was gonna say, I also didn't know that you were pre-med also. I followed in that exact same path as you did, because that's what, being of Asian descent, that's where you go. But thankfully I was like you and discovered paleontology also, so I didn't know that was your background. That's really cool.
Yep. I think that story is much more common than people think, and I wish someone had pulled me out earlier, because I was miserable. Like...
Oh yeah, like I finished and I took the MCATs and everything, and I was like, there was like a day where I was like, I don't want to do this.
Yep.
And then I moved on.
Yep. I had that day when I was studying for the MCATs, and I'm like, I have to study chemistry again? Like, no, I'm not doing this.
Oh my goodness. Well, yep. I'm very glad you both ended up in paleontology.
Yeah.
Alrighty. So here are some questions from our chat. There's lots of good questions in here. As always, we'll try and get to as many as we can. So let's see. Oh, why are we not crabs? That's a great question from Lucas. First, let me do another question, then we can get on to the eternal question of why are we not all crabs? This is from Cavaramus. What is responsible for the continuous replacement of teeth in animals like sharks? What mechanism stops it in most mammals?
Ooh, that is such a good question. Okay, so basically we have this thing called dental lamina, and that's a fancy word for a little bundle of stem cells that lives at the end of each tooth. And so in mammals, that little bundle of stem cells stays active from the time you're born till basically the time that you develop your last set of teeth. In us, that's our adult teeth. And then that little bundle of cells, for some reason, senesces, meaning gets old and dies. And then we get no more teeth. And we're stuck with the pair that we have. That's it. In sharks and a lot of reptiles, that little bundle will actually stay. And as the jaw moves or grows, it will move and then make the new tooth. And the new tooth will push up against the old tooth and replace. And therefore, a bunch of reptiles and a bunch of sharks and fish and almost everything else keeps replacing their teeth throughout their lives, but we don't. Now, I'm just going to add a thing that I know you didn't ask, but a lot of people say, why don't we just, find a way to keep those cells in us alive, and then I don't have to get dentures or I don't have to get implants or whatever. Why don't we keep replacing our teeth? First of all, that would be a nightmare. Second of all, if you actually keep those cells alive, that's what causes cancer in humans. It seems like somehow we don't have the mechanism to stop those cells from over-reproducing and causing odontomas, which are basically tooth cancers or tooth abnormal growths, not always cancerous. So in mammals, for some reason, early on, our ancestors have basically shoehorned themselves into having just the two generations. Now, don't be too sad about it, because our teeth are actually quite a bit stronger than a lot of reptile teeth. Our enamel is thicker, our dentine is thicker. We also have a bunch of ways to heal internal dentine and replace that. And of course, modern medicine makes it easier. So it's not too bad. If you have to replace your teeth every 3 days, like, imagine the pain.
And just the teeth, like the shed teeth everywhere, like
There's just coughing in the middle of a meeting.
Yeah, oh no. Sorry, this happens all the time.
Yeah.
Oh, Gabe, we can't hear you.
I said that maybe we could have tooth jar, but then I regretted saying it, and I probably didn't need to repeat it since my microphone was off. So
Your
anyway, move on.
microphone knew. Your microphone knew. You're not
Uh,
gonna.
nice.
Uh, let's see. So if you would like, we can go into the question of why are we not all crab from Lucas.
Why are we not all crab? I would say that it was an evolutionary mistake, realistically. I mean, really, it's just who we're related to. And unfortunately, somewhere down the line, the group of cells that decided, hey, we're going to go and make invertebrates that way, and the group that decided to make vertebrates that way just couldn't reconcile. And it was a mistake. I agree.
Yeah.
We should all be crab.
We should all.
I could have natural armor. Just be like, come and get me. I'm good.
Yeah. This is really depressing and unfortunate for all of us, for humanity as a whole. This is from Mermaid Labs, saying, sorry, you probably already said this, but when approximately did osteocytes come into being?
Ooh, good question. I did not actually put a specific date on that. And so, to our knowledge, it's around 420 to 450 million years ago. Those are the jawless fish that we know have osteocytes. But realistically, we haven't cut every jawless fish that is out there. And it turns out that some early fish have things that look like osteocytes at the base of their teeth. But so much crazy stuff is going on down there in the Silurian and Devonian that it's so difficult to tell, is that an osteocyte or is that a new cell that they had invented at that time that went extinct later? Because all we're going on is the shape. So that's actually one of the things that I'm trying to address with my PhD when I can get back to it, is we're scanning a bunch of these cells and seeing if we can actually tell by shape alone if it's the same cell type or not, because osteocytes can be all different shapes. And so assuming that everything that is that shape is an osteocyte is kind of a circular problem, right? Because then you start to assume, oh, everything's an osteocyte. But at that time, there was so much experimentation that it wouldn't be insane to have all these different cell types as well. So 420, 450 is pretty safe, but realistically, we're not 100% sure.
This is so cool that you're,
I
like,
know.
as a paleontologist, you're studying cells and, like, things that, you know, growing up we were taught, like, you don't preserve that kind of information in the fossil record. But, you know, like you said, it's the golden age of paleontology, and, you know, this is something that we can learn about from, like, tiny microscopic things, and, like, have all these connections that we never knew we had before. Is that something that, like, blows your mind sometimes when you're doing your own research?
Only every day. And the day that stops happening, I'm leaving paleontology. I promise everybody. Like, for example, and I can't talk about this too much because of embargoes and blah, but I have a paper that's coming out later this month that's basically showing, so these osteocytes, right? The best way we've been able to image them for a long time is histology, like I've shown you guys, or scans, where they're basically these blobs because our scans were not high resolution enough. So I got together with a bunch of these physicists and material scientists that do all this kind of scanning that on batteries and even smaller things. And I was like, hey, what if we do this with fossils though? And we got to scan them and we got ridiculous images. Cool. And I'm so excited to share this with everybody. But we don't only live in the golden age of paleontology, we live in the golden age of other sciences. And now everyone's connecting so much more easily. And I think there's going to be a lot of overlap, and we're going to see some really cool things in the coming years.
That's awesome. I love being a scientist.
I know. I'm just getting to hear about this kind of stuff. So yeah, everybody keep an eye out for Yara's paper coming out, because that sounds like it's going to be cool. Alrighty, more questions. There were lots of good questions. Inspired some cool dialogue here.
I want to shout out to John Friel really quick, who says reject crab, return to worm in the chat.
Oh, that worm was very interesting, but also, ooh. Okay. This is another question from Cameroonians. One of the diagrams showed that in cartilaginous fish with fish with cartilage. Um, there are actually some bone in their skeletons. What parts are ossified?
Good question. So, basically, a lot of people. So let's talk about sharks for a moment. First of all, sharks, real cool. Second of all, people think of sharks as like the primitive version of bone, right? Because they have a cartilaginous skeleton. But that's not true at all. Sharks actually reverted to cartilage. They looked at bone and they were like, yeah, no, not for us. And they went back. So actually, shark ancestors have almost fully ossified or partially ossified skeletons. And then their ancestors were like that. Sorry. And then their descendants were like, no. And this actually happened in fish several times. Like sturgeon are like, yeah, bone on the outside, but not on the inside. So there was a lot of experimentation when it comes to that kind of stuff. So early ossifications in cartilaginous fish, when I showed it in that diagram with sharks, I was referring a big part to their ancestors, which are almost completely ossified. Some have around their vertebral column. Some have in their scales they're ossified. Also, the bases of shark teeth have something called an osteodentine, which is like bony dentine, so that also counts. And that's still around today. So like modern sharks, if you see like a really big like megalodon tooth or whatever, that weird spongy base, that's a mix of dentine and basically bone. It's a really interesting, very specific tissue that sharks basically do. So there's lots of ossification down the line in cartilaginous fish.
Interesting. I love that
I
they
like
were just
shark.
like, yeah, like,
Yeah,
no
bones for us.
bones for us. We don't need those. Oh, this is a good question. This is from Noah. Would you? They found a dead fox in the woods. Do you have any recommendations on how to collect bones and any tips on finding more dead things?
I love these questions. Okay, so initially I was a birder, and so I would look up all the time, right? And so then you realize there's a lot of really cool stuff on the ground. So my advice for birders was always just stay looking up. But if you're looking for bones, keep looking down. So it sounds really simple, but really just look at like the bases of trees. Foxes, raccoons, squirrels always tend to like die around bases of trees. I think is just some partial shelter, and they kind of die there. Check your nose. You're gonna get real familiar with the smell of death, and some people walk away from it. That's not us. Okay, everyone in this chat, no, you walk towards that. You find a stick and you poke it. So for collecting bones, I would first and foremost tell you to double check your local laws, because things like migratory birds, don't touch them. Probably a mistake. Marine mammals, depends on where you are. Most places, California, don't touch them. What else? Always have a couple of trash bags on you. That way, you know, if it's still a little goopy, you don't want to touch it, fine, be you. And you can just pick it up kind of like a dog poop. And then you can clean bones pretty easily in many different ways. If you have a backyard, just put it on an anthill if you have one. Ants are your best friends, especially if it's anything with small, delicate stuff. You can also do a very dilute hydrogen peroxide mix. So if you have a bucket, you just put a little bit of hydrogen peroxide, you soak it, don't smell it, don't lick it, but yeah. And there's a lot of resources online on how to clean bones and properly display them. Most mammals, like raccoons and foxes and stuff like that, really okay, super easy. Yeah, but definitely double check your local laws so you know you're not in too much trouble.
I love if anybody takes anything from this, the don't look the bones tip, I feel is especially important.
Yeah.
I think you're muted again, Gabe. Your microphone heard you were talking about crabs and was just not willing
No,
to go
I
through
wasn't
that.
talking about crabs this time. I just remembered I have a pig skull buried in my backyard that I forgot to look at.
You just remembered that, huh?
I did, and we just
months ago.
Yeah, no, like a couple years ago, and now it's covered in rock and astroturf. So now I gotta figure out where it was.
I think you should just leave it. It's a future fossil.
Oh yeah, I'll do that for the next people who move in.
You know, I was about to say, whoever buys your house after you is going to have a bit of a surprise.
But they could have a future scientist who finds the pig skull and be like, "Oh, what's this?"
Or they're going to think your house is haunted. Either
I mean,
way,
that's cool too.
it'll help with it.
Whenever you're done with your skulls, always put them in walls for your next tenants. It's kind of a rule.
Yeah.
Okay, guys. I think this is going to be our last question. There are lots of other good questions. Thank you, guys. I think everybody really loved this talk, but I think this is a good question because it's for a student. This is from Jeremy. He says, "I'm thinking of using histology for his master's thesis. I've got a textbook coming in the mail, but could you give him some examples of what histology can tell them about a fossil other than its age?"
Absolutely. And I think I might know which Jeremy this is. And good for you. Keep using histology. You can always ask me about histo. So histology, you can do a bunch of things aside from age. You can actually check not just growth patterns, but you can check like metabolism. So like, does an animal have a high metabolism or a low metabolism? And now there are ways to actually quantify that. So one of the pictures I showed had circles within circles within circles. And basically what that is your vessels pushing through bone really quickly to resorb bone and deposit bone really quickly. So some animals will almost have none of that. Like a lot of reptiles, slow growing, they don't actually take out a lot of bone and re-put in bone. But things like dinosaurs and birds and some mammals have really high bone turnover. And that can tell you how that animal is kind of where they are in the metabolically active range. And that can be important for life history, for behavioral implications. So you can tell a lot about metabolism. You can obviously tell a lot about osteocytes. Like for me, you can look at cells. You can also look at cell size. So one of the things that definitely blew my mind is if you look at an osteocyte lacunae, the cave that the osteocyte lived in, you can measure how wide or was the volume of that lacunae, and from there, you can use that as a proxy for the size of the cell. Now, amazingly, these cells track with genome size really well. So you can actually estimate how big certain animals' genomes are. Extinct animals, we can actually figure out how big their genome was. Now, it doesn't actually tell you what's in the genome, but it tells you how big the genome is. And that's important because animals with fast metabolisms, like hummingbirds and whatever, have very small genomes. And some animals that can deal with long hibernations and grow slow, like salamanders and lungfish, have giant genomes. So then some of the questions we have is like, well, where did therapsids fall in that? Where did mammoths fall in that? How big is their genome? What kind of lifestyle did they have? That can all be answered with histology, which I think is amazing. So, hope that helps.
That is awesome.
That's cool. That's so neat.
Yeah, Yara, I'm in awe of how awesome you are as a science communicator. So like, I bow and just be like, thank you so much for being on our show today. You were fantastic.
Yes, thank you.
Thank you so much. And for anyone that still has questions that are left unanswered, you can hit me up on Twitter. I posted about this talk specifically, so just comment under that. I will be answering them from bed the rest of the day. So feel free to send those over. And thank you so much, you two, for having me and for running this all pandemic long.
And hopefully, that means we're going to keep going because, Yara, today is our 50th episode, as Brittany said, and that makes you our 50th guest. So thank you all so much for joining us on 50 episodes of Fossil Friday Chats. We started this as like oh no, what are we going to do when our museum's closed? And it kind of became something, that we love to do, and we're hoping to continue forward. So yeah, thank you all so much for watching.
Thank you.
Yara, if somebody wants to check out your blog, where can they go as well?
It is thebarebones.org. It is a little bare right now, but we'll be soon. So
all right. Cool. So we'll make sure we put all that in the chat. And, once again, thank you all so much for joining us. If you like this program and want to support programs like it at the Alf Museum and the Western Science Center, you can find links on how to do that in the description below. And as always, please like and subscribe to our channels so that you can hear more stories from the world of paleontology. And we will see you all next week. Bye, everybody.